أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم فأقم وجهك للدين حنيفا فطرة الله التي فطر الناس عليها لا تبديل لخلق الله ذلك الدين القيم ولكن أكثر الناس لا يعلمون صدق الله العلي العظيم. As I mentioned in previous class, the rate of enzymatic reaction depends on substrate concentration, and the curve describes a hyperbola. At low substrate concentrations, the initial rate of enzymatic reaction increases rapidly and follows a first-order kinetic. It means that the amount of product formed is proportional to the substrate concentration. However, as substrate concentration increases, the reaction saturates and reaches a point of equilibrium in which the rate of enzymatic reaction no longer depends on substrate concentration. At this point, due to excess of substrate, all of enzyme molecules are in the form of enzyme-substrate complex, and the maximum rate of enzymatic reaction is reached. I also mentioned that the Km is the substrate concentration at which the rate of enzymatic reaction is half of the maximum rate of that reaction. Actually, the Km is the substrate concentration at which half of enzyme molecules are forming enzyme-substrate complex. The smaller value of the Km, the higher activity of the enzyme and greater apparent affinity of enzyme to substrate molecule. Today, I want to talk about enzyme inhibition. Enzyme inhibition may be reversible or irreversible. In reversible enzyme inhibition, the inhibitor binds to enzyme reversibly by weak intermolecular bonds like hydrogen bonds or van der Waals bonds. Irreversible inhibition involves either the denaturation of the enzyme or formation of covalent bond or a highly stable non-covalent bond with enzyme. The examples of irreversible inhibitors are organophosphate chemical warfare agents known as nerve agents which inhibit cholinesterases. There are two major types of reversible inhibition: competitive inhibition and non-competitive inhibition. There is also another inhibition which is called an uncompetitive inhibition, which is a specific type of non-competitive inhibition. In competitive inhibition, the inhibitor has a similar structure to substrate molecule, and so they compete to bind to active site of the enzyme. By increasing the concentration of inhibitor, the substrate molecule is displaced from the active site of enzyme, and so the enzyme activity is decreased. By increasing the substrate concentration, the inhibitor is displaced from the active site of enzyme, and so the inhibition is decreased. Please pay attention to respective Michaelis-Menten and Lineweaver-Burke plots. In competitive inhibition, the Km is increased, but Vmax is unaffected, as you see in a Michaelis-Menten plot. The blue curve represents the enzymatic reaction rate in absence of inhibitor, and the red curve represents the enzymatic reaction rate in presence of a competitive inhibitor. As you see, by increasing the concentration of substrate, the maximum rate of the enzymatic reaction is reached. Here, the Km of enzymatic reaction in presence of inhibitor is greater than Km of that reaction in absence of that inhibitor. Both curves reach to the maximum rate of the enzymatic reaction. Please pay attention to Lineweaver-Burke equation. Vmax is unaffected here and is constant, but Km is increased. So this ratio, namely Km divided by Vmax, which is the slope of the line, is increased, as you see in red line. So the slope of the line in presence of competitive inhibitor is steeper than that of in absence of competitive inhibitor. In non-competitive inhibition, the inhibitor has not similar structure to that of substrate molecule. Also, the inhibitor has a different site of action than that of substrate molecule. So, by increasing the concentration of substrate, the inhibition is not decreased. Please pay attention to respective Michaelis-Menten and Lineweaver-Burke plots. In non-competitive inhibition, Km is unaffected and Vmax is reduced. Please pay attention that the Km in presence of inhibitor is equal to Km in absence of inhibitor. And the Vmax in presence of inhibitor is smaller than Vmax in absence of inhibitor. And so by increasing the concentration of substrate, the maximum rate of the enzymatic reaction is not reached in presence of inhibitor. Please pay attention to Lineweaver-Burke equation again. Km is unaffected here, but Vmax is reduced. So this ratio, namely Km divided by Vmax, is increased. Again, the slope of the red line is steeper than the slope of blue line, namely the slope of the line in presence of a non-competitive inhibitor is steeper than the slope of line in absence of a non-competitive inhibitor. A specific type of non-competitive inhibition is uncompetitive inhibition. In this type of inhibition, inhibitor only binds to bond enzyme, namely inhibitor only and exclusively binds to enzyme substrate complex. After binding of inhibitor to enzyme substrate complex, no product is formed. Also, the inhibitor structure is not similar to that of substrate. Please pay attention to Michaelis-Menten plot of this type of inhibition in a slide. As you see, the Km is reduced and also Vmax is reduced. Please pay attention to Lineweaver-Burke plot of this type of inhibition. The red line represents the enzymatic reaction rate in presence of an uncompetitive inhibitor, and blue line represents the enzymatic reaction rate in absence of the inhibitor. Because the Km is reduced and also Vmax is reduced, the ratio of Km divided by Vmax will be constant. So the slopes of red line and blue lines are equal. So these two lines are parallel. Question. Please pay attention to Michaelis-Menten plot of uncompetitive inhibition. As you see, both two curves cross each other. Please answer that why they don't cross each other in Lineweaver-Burke plots. والسلام على من اتبع الهدى.